Here we have an illustration of periodic selection with some emphasis on genetic hitchhiking and mention of selective sweeps. We have a population. It consists in part of these individuals that have the genotype little a, big B. We don't actually see in this figure what the rest of the population looks like, but for the sake of understanding periodic selection, let's assume that the little a allele is potentially making up the entire population, but that the big B allele is only found within this subpopulation. And in fact, we're keeping track of the frequency of allele B, and what this is showing is that allele B is present within this population at a low level. Now, for periodic selection to work, what you need are clonal populations. As a consequence of clonality, there is a linkage between the A locus and the B locus, and this is true no matter how separated within the same genome or on the same chromosome the two loci are. Now, within this little a, big B genetic background, we have a mutation that converts the little a allele to a big A allele. This big A allele displays a selective benefit relative to the little a allele, and it's a sufficiently large selective benefit that the entire genome that the big A allele in displays a selective benefit relative to the rest of the population. As a consequence, there is an increase in the frequency of the big A allele. This is our selective sweep. This is periodic selection, increase in the frequency of this particular clone that happens to have associated with it this beneficial mutation big A. But in the course of the increase in the frequency of the big A allele as found within just this genotype, you also have an increase in the frequency of the big B allele, only because that big B allele is linked to the big A allele, and thus you see an increase in the frequency of allele B. If the allele A goes on to the point where it has become fixed within the population as a consequence of carrying the allele B along with it, allele B too will become fixed within the population. And this is true whether or not big B allele supplies a benefit, supplies no benefit, or even is a detriment, so long it is not so much of a detriment that the genotype that is holding the big A allele is not more fit than other genotypes within the population. The big B allele, in fact, will be carried to fixation within that population.